Hello everyone and welcome back to Gurum's classes. It's already 8:20 p.m. in the evening. In fact, here is night. What we can say is then also okay. Yesterday also I was making the video in the same time. Okay, today, in fact, in this evening we'll be doing is experiments on photosynthesis. Now, experiments on photosynthesis means how will we prove that a plant has photosynthesized? Okay, a certain plant has photosynthesized. How will we come to know? Okay, or various factors are there, like we have is carbon dioxide, we have is oxygen, we have is sunlight, we have is chlorophyll. All these things are present in together, okay, to conduct the phenomenon called the process called photosynthesis. Okay, so based upon that we will come to know what are those factors and how are they necessary. For example, number one is starch is produced as the end product of photosynthesis. How will we come to know that yes, it is the starch only? How will we come to know? Okay, so what is done basically is, first of all, because we have studied that 6CO2 plus 12H2O, we write is chlorophyll, sunlight, and we say that glucose is produced, and that glucose after polymerization, starch is produced. Now that theoretically we have come to know. Okay, fine. Now how will we prove that starch is produced at the end of photosynthesis? So in that case, this is the experiment number one, and I have written the experiment number one in a flow chart, and you have to remember these steps. Once you remember these steps, all the other experiments, okay, the same steps will be followed. Only few things will be changed. Rest will be same only. Fine. So first of all, number one experiment is starch is produced as the end product of photosynthesis. So what is done? First of all, try to understand. A potted plant is taken. What is taken? A potted plant is taken. Now the potted plant is de-starched. That means it is kept in a dark room. Please. Okay, which room? Dark room. So here you have to mention dark room. The potted plant, you have to take a potted plant and you have to keep that potted plant in a dark room away from the sunlight for minimum, okay, 48 hours. When you keep the potted plant in a dark room 48 hours, what happens is that, whatever starch was present in that plant, the plant will utilize within 48 hours for its living to survive. So that means when you take out the plant after 48 hours, there will be no any starch left in that potted plant. So that means, okay, now we can, be assured that there is no starch. Fine. After we conduct the experiment, if we find starch, then we can say that yes, that means, okay, the starch, that is present in the plant is after the experiment. Before the experiment, the starch wasn't there. Why? Because the starch was already, the plant was already starched, or the starch was already utilized by the plant. Yes or no? Within 48 hours. So this, okay, that is why the question might come, why do we de-starch a plant, okay, to conduct an experiment upon photosynthesis? We de-starch a plant, okay, before conducting any experiment upon photosynthesis to ensure, or to ensure that whatever starch is produced at the end of the experiment is produced through the experiment only, through photosynthesis, to ensure that the plant is de-starched, or to ensure that the plant which we are taking, the plant which we are starting with, is without any starch. Fine. So we take a potted plant and then we keep the potted plant in a dark room for 48 hours, and then after that we keep the plant now in sunlight. Yes or no? Now we keep the plant in sunlight. So we keep the plant in sunlight for 2 3 hours. After that we pluck one leaf, okay, and then we boil that leaf. Okay, we take one green leaf and then we boil that leaf in methyl alcohol or methylated spirit. We boil that leaf in methyl alcohol. When we boil the leaf in methyl alcohol, chlorophyll is removed, but the leaf becomes crispy. So that is why that crispy leaf is then put in warm water and it becomes soft. Fine. Now chlorophyll is already removed. Now when it becomes soft, that leaf, then that leaf is taken, okay, and what is put? Iodine is put. Now in that leaf, what is put? Iodine is put. When iodine is put, then what happens is that iodine in presence of starch, iodine is brown in color. Okay, and when iodine in presence of starch, that brown color changes into bluish black. You can try this at home also. Take iodine, okay, and then you cut a potato, and then you can put some drops of iodine there. Then the potato color changes to bluish black. So this leaf, okay, now upon this leaf, if iodine, when iodine is put at the last, okay, then the iodine color changes to bluish black. If the color of the leaf changes to bluish black, we can say that in the leaf starch is produced, and this starch must have come from photosynthesis, the experiment, okay, that because the plant was kept in sunlight. Why? Because in this leaf beforehand, when it was de-starched, there was no any starch present in this leaf. Okay, after that, okay, when it was kept in the sunlight, okay, and then the leaf changed into bluish black color, proving that starch is present. Okay, so this experiment might come in your examination in section B. Now another experiment we have is sunlight is necessary. Again, same process is there, only few things are okay changed. We take is a potted plant, same. We keep the potted plant in a dark room for 48 hours. The plant becomes de-starched. Then, okay, we take out the potted plant in the sunlight, and of a leaf, suppose we choose this leaf. Now we cover a section of this leaf, okay, with a paper or with anything. Okay, the main motto is to block the sunlight. We block this section, you can use any shape paper also. So this region is blocked from sunlight. Okay, we put some clips, okay, and then this region of the leaf is devoid of sunlight, and we keep the plant, potted plant, like the experimental setup, in the sunlight for 2 4 hours. After that, this leaf is taken, this leaf is taken, and then it is boiled in methylated spirit. Same again, the leaf is boiled in methylated spirit. Chlorophyll is removed now, and then what happens is that it is put in warm water. The crispy leaf now becomes soft. Then iodine is put. When iodine is put, what happens is that here in this case, this is the leaf, okay, and this in this region, okay, this region was blocked. So as a result, the bluish black color will be seen here in this region, okay, because this exposed region, the bluish black color will be seen in this portion. Okay, by dropping iodine, bluish black color will be changed in these two regions, but not in the middle section. Why? Because the middle section did not receive sunlight, because the middle section was prevented from sunlight. As a result, okay, no photosynthesis took place here. No photosynthesis means there was no any stars produced. That means sunlight is necessary for photosynthesis. This is the experiment. Next is chlorophyll is necessary. Now here we take is a special type of plant, a plant which has variegated leaf. You can find variegated leaf, okay, having plants. Variegated leaf means a leaf in which the whole section of the leaf, the whole blade of the leaf is not green. Okay, some sections are green, certain sections are green, and rest could be yellow. Such plant is taken. Okay, now the variegated leaf potted plant is kept in a dark room for 48 hours, okay, to ensure that the starch is absent in the plant. Then after that, the plant is kept in sunlight. Okay, now what is done is, before keeping the plant in sunlight, what we do is we take the leaf, and then what we can do is we can draw the leaf size, okay, in a graph paper. Also, what we can do is we can take a graph paper, and we can mark. That also can be done. Okay, this is the graph paper, and by taking one of the leaf, normally, okay, see, yes, okay, a leaf is drawn on a graph paper to mark the positions of the green portion. Then the plant is kept in sunlight, and then photosynthesis take place. After that, okay, a leaf is taken, the variegated leaf is taken, and then that variegated leaf is put in alcohol, and then it is put in warm water, and then iodine is dropped. When iodine is dropped, only those portions will show change in color from brown to bluish black, which was green in color, or in, okay, so that means here also in this portion only these regions will show bluish black change in color. Okay, the rest of the portions do not show any change in color. Why? Because chlorophyll was absent in that region. Here also it proves that chlorophyll is necessary for photosynthesis. The rest of the region does not show any change in color. Why? Because chlorophyll was not there. As a result, the starch was not produced there. Next is carbon dioxide is necessary. Most of the time this experiment comes. Okay, carbon dioxide is necessary. Again, the same experiment. What is there? We take as a potted plant, we keep it in a dark room for 48 hours. We take the potted plant outside in the sunlight. But what is done is we take a leaf, and then half of the leaf is inserted in a conical flask which contains potassium hydroxide. Now try to understand this. Okay, this is potassium hydroxide. The function of potassium hydroxide is, okay, it absorbs carbon dioxide. Now potassium hydroxide absorbs carbon dioxide. As a result, what happens is this the conical flask inside had carbon dioxide or had air, and that carbon dioxide, instead of entering the leaf, potassium hydroxide absorbs, carbon dioxide. Now what happens is that the outer portion of the same leaf can easily receive carbon dioxide from the air. Now the experiment is kept in the sunlight for 2 4 hours, and then afterwards this leaf is taken and it is boiled in methylated spirit. It is warmed, okay, in warm water. It's made soft, and then afterwards iodine is added. Then what happens is that, okay, then what will happen is only the region which was exposed outside in the sun will show bluish black color change. Okay, and then the region which was inside will not show any change in color. Why? Because here starch was not produced. Why? Because to form starch, carbon dioxide is necessary, and carbon dioxide was already absorbed by potassium hydroxide in this conical flask. As a result, this region did not photosynthesize, or starch was not produced only in this region, proving that carbon dioxide is necessary for photosynthesis. Okay, so that means all these steps are necessary. Now another experiment is there, that is oxygen is produced as a byproduct in photosynthesis. So here we take is an aquatic plant. Okay, so here we have taken is hydrilla. Hydrilla is an aquatic plant which is found under the water. Okay, and it can, photosynthesize with the sunlight, and oxygen gas is produced. Okay, so a beaker is taken, and in the beaker water is put, and hydrilla plant is taken, and then it is covered by a conical flask, and we have to take is a test tube. In the test tube we have to put is water, and then we have to insert it on top of the conical flask. Then we have to keep the experiment in sunlight, because as you know very well, photosynthesis means sunlight. After keeping the experiment, okay, in sunlight, what we will observe is slowly, okay, bubbles of air will come out. Okay, and then the air will be collected on top of the test tube with the lower displacement or downward displacement of water. Later on, okay, the test tube is taken. Okay, and then when we keep the test and then, if we keep a burning splinter near the test tube, the burning splinter will glow more vigorously, because oxygen gas helps in burning. This proves that the gas that was collected on top of this test tube is oxygen gas. Okay, so this experiment also comes. This also, all these experiments are important. All these experiments from photosynthesis chapter comes. Okay, so as you know very well, in the previous video we had already discussed that photosynthesis is very important for food, for oxygen. Okay, so plants also need to photosynthesize, and for that reason plants, they adapt themselves in various ways so that they can photosynthesize in the best possible way, so that more and more food could be produced in them. So various adaptations are there, that is to have thin leaf. Thinner the leaf, more amount of sunlight can pierce to the depth of the leaf. So this is one adaptation. Another is broad leaf. Broader the leaf, more is photosynthesis. Or more stomata. If more stomatas are there, better is transpiration. Better is transpiration and better is what? Exchange of gases. So this also is one of the adaptation plant. They adapt themselves. They bring change in them. More chlorophyll, greener the leaf, better is photosynthesis. And the last one is more venation. More venation, more water, transportation is there, better is photosynthesis. So question might come, right? Few of the adaptations, okay, of plants for photosynthesis. Then you have to write these experiments. Okay, students. So at last, kindly study carbon cycle. Carbon cycle means after photosynthesis, carbon dioxide is produced, carbon dioxide is given out by human beings, but that is taken by green plants. Okay, animals also produce carbon dioxide, that is also taken by green plants. Combustion, okay, all these industries, factories also produce carbon dioxide in the air, which is also taken by green plants, and then green plants gives us oxygen. And then plants, when they die, okay, like suppose wood logs are there or fossil fuels are there, all of the fossil fuels, it also produces carbon dioxide. Okay, so in that case, carbon dioxide is produced by all human beings, all animals, industries, factories in the air, that is taken by plant and is converted into oxygen. Okay, so this way carbon, okay, enters to the green plant and becomes oxygen. Or we human beings again give carbon dioxide, in return we get oxygen from plants. So carbon this way forms a cycle. So kindly go through carbon cycle. Few questions from carbon cycle also might come. So this way, dear students, we end up the chapter on photosynthesis. Okay, so thank you very much.